This is the second lecture series of the course Computer Vision. Today we'll talk about biological vision. This topic is an extension of our previous lecture, which is about introduction to computer vision, where we see both computer vision and human vision in high-level manner. But today we'll dive more on biological vision. In the next session, we'll talk about camera systems, which is more to the baselines of computer vision. The two most important topics that we will be talking about today are how do we see, the historical theories of vision, and also we'll talk about biological vision, the theories and results behind, the psychological and cognitive neuroscience of vision research findings. The sources for this lecture are partially the Cognitive Neuroscience of Vision book, which is written by Marta Farah, the Foundations of Vision, which is written by Brian Wandel, and the Natural History of Vision, which is an MIT Press published book, which is written by Nicholas Watt. So let's start by discussing the historical view of vision. In order to understand vision, we need to have understanding of the physics of light, because we are perceiving light, right? So the first requirement for understanding vision is understanding the physics of light and its interaction with objects. The second one, understanding optics. We have to understand optics. And the third one is we need to know how the brain works, right? But generally, historically, the view of vision has been discussed from two different dimensions or perspectives. The first one is extramission, and the second one is intromission. Philosophers who have been advocating for extramission says the human eye is a very powerful entity or component of a human system that emits lights to the external objects, and the reflection of those lights will be further processed by our vision system for us to know what is what or what is where. But when you come to intromission, advocating philosophers, they say the human eyes is not some supernatural entity that emits light to the external world and absorbs it back, but they define our vision system as a system that absorbs lights from objects in the environment and process it after the process called refraction of lights. Let's discuss each of these in detail. As I have already discussed extramission theories of vision, these people are like Plato is one of them, Euclid is the other one. So they say eye emits rays and a person perceives the objects struck by these rays. Specifically, Plato said, from our eyes flows a light similar to the light of the sun. So this theory of vision has some spiritual aspects. It's not that scientific, but it has been supported by very renowned philosophers like Plato and Euclid. So the question here would be how that interact with luminous sources like sun and other luminous sources like moon. For instance, are we emitting that powerful light to moon and sun so that we can get, that powerful luminous intensities back to our retina? So many people from the intromission perspective question or raises questions like this. So for instance, the first theory, from this perspective is Ptolemy. Claudius Ptolemy is another philosopher who lived around 168 AD. So he said, or he advocates for the concept of refraction, but initially his idea is mainly based on extramission theory. So he says there is visual flux for our eyes, an eye that emits light to the external environment, the light that gets reflected by an object, and a human eye system again that understands the refracted form of these two combination of lights. So basically, Ptolemy is also used to refer to concepts that advocates for the refraction theory. That means objects are created by refraction of lights back to our retina. So this part of this theory, like specifically the refraction part of this theory, is very common to the people who are advocating for intromission. But the problem is that extramission theorists say the human vision system is an active vision system. That means it has its own light source. But the intromission guys say no, the human vision system is passive, so it cannot have its own light source. So therefore, according to Ptolemy, visual flux from our eyes, external light and instead of refraction, produces sight or vision in general. So the cause of sight generally is threefold, as they claim. The first one is the light of the innate heat passing through the eyes, which is emitted from our eyes. The second one is the principal. The second one is the exterior light kindled to our own light. They're assuming some kind of fusion between our light and the light of the external entity that we are focusing on. And the third one is the light that flows from visible bodies, for instance, flame or color. So they say without this, the proposed effect of vision cannot occur. So when we further discuss extramission theory, they face many difficulties. The first one is compromised, or their theory is mainly compromised by this question. For instance, why do we see faraway objects instantaneously when we open our eyes? If we have to emit light, and if that light has to be reflected back to our eyes, then there should be some kind of latency, right? But the reality in our vision system is not like that, because when we open our eyes instantly, we see something. So the reason for this given by the extramission theorists is that the visual speed that leaves the eyes is exceptionally swift. So they advocate for the exceptionally swiftness of our vision system. So they try to hypothesize the possibility of that instant response from the external environment, even though the human eye is emitting lights. The second question that tries to debunk the theory of extramission theorists is why don't the vision systems of different people looking at the same object interfere with each other? You see, if you are defining the human vision system as an active system, and if every human eye emits light, this light is physical energy, then why do these physical energies why do not these physical energies interfere with each other? Their answer for this is not sufficient. The other one is, what if the eyes are closed when the visual spirit returns? For instance, if we are emitting lights to the target object, and if we instantly close our eyes before the return of that reflection, then will we be able to see something? And the answer for this one is more related to the spiritual aspect of human vision system. They might raise, concepts related to evil eye, the third eye, and something like that one. So because of this, we could not use this theory of vision for scientific research. But when we come to the second vision of a view of vision, it's intromission theory of vision, which is mainly advocated by Aristotle, Democritus, and different people. So basically, this intromission says that the external objects are responsible to emitting lights, and the copy of that emitted light is transported to our eyes through the atmosphere, and our eye uses some components like retina to refract these lights and make some kind of semantic image or objects. This is also questionable. Later it has been improved, but the foundation of the current computer vision scientific society is intromission theory of vision. The other guy who tried to challenge the extromission theory of vision is Abu Ali al-Hassan ibn al-Hassan ibn al-Haytham, or mercifully he is shortened to al-Hazm. He's a very, the greatest optical scientist of the Middle Ages. He's Iranian. He wrote this book called Kitab al-Manzir, or Book of Optics. Um, he says generally light is a physical phenomenon, which is true, and he dissects or he distinguishes light from vision. He says light radiates from self-luminous bodies and moonlight. He's more to intromission, and he says light travels in straight lines. And when you talk about the way light radiates and the way light transports from one place to another place, he also talks about the concept of medium. Uh, that means the medium that light hits. The first medium is defined as transparent, that lets refraction of lights, and the second one is opaque, which doesn't allow transmission of light, so it mainly allows reflection of light. So he defined medium of objects in a computer vision system as transparent, which allows refraction, and opaque, which is uh, more to translucent, or an entity or an agent for reflection. So he says light is refracted between two transparent mediums, the first type of medium. And when light hits an object, it radiates in all directions. And he advocates for, again, pointless theory of vision. So he says we see a collection of points on the surface of objects. And again, from the geometric theory of vision, which defines the one-to-one correspondence between the word and the image formed in our eyes. That means whether we are seeing one object that exists in the real environment the same way it is represented in the real environment inside our eyes, or whether we are reconstructing the real world correctly or not. So this guy, Al-Hazan, has been talking about or writing about these concepts mainly. He's intromissionist, um, and he also advocates for the theory of refraction. He believes images are created in our vision system by refraction, and the refraction happens in the medium called transparent medium. So usually we call that transparent medium retina. That being said about Al-Hazan, let's talk about how lens operates and image formation happens. As Al-Hazan said, light hits some objects, and its ray refracts or reflects to all directions, but the ray of light leaves the light source and travels along a straight line. That's one point that we have been discussing about. But when light hits an object, and the next step would be either reflection or refraction. That means when the light hits either transparent or opaque objects, it will change its direction. For instance, if it hits transparent object, it refracts, and when it hits translucent or opaque objects, it reflects back to its origin or some other direction. So this diagram to the right shows how light travels in the atmosphere and how it behaves when it hits the surface normal. Here we have a surface normal here, and the incident ray is a ray or the source light. And when this source light is emitted from one object in the surface and hits some surface normal, some target object, most of the time in human vision system it's going to be the lens. Depending on the nature of the objects being hit by that light, it might refract or reflect. When the object being hit is transparent, like lens, the light or the incident ray will be refracted and it will become refracted ray. But when it hits translucent or opaque objects, it gets reflected back and it creates reflected ray. So if the object is our lens, for instance, as we already said, then the useful light for imaging is the refracted light. So this is mainly preached by the intromissionists and also by Al-Hazan. But when we see the concept of refraction, the theory that is brought by Alhazen, and the concept of extremissionists like Ptolemy knew, the phenomena of refraction, because they said there is a concept called refraction after after the light is being emitted by the human eye and gets reflected back to the human eye. So this concept of refraction unites the three theories of both Ptolemy, Alhazen, and the general refraction theory. But the problem with the Alhazen's you know, the theory has been since light from a surface point reaches the entire surface of the eye, how is it that we see only a single image of a point? Because Alhazen took the assumption that when the light gets emitted and hits some kind of object, then the reflected or refracted light goes in a straight line in all over the directions in the environment. So if light irradiates in all directions, how is that possible for us to see a single image of a point, given we have a light source or a light or a set of lights irradiating in all directions in the environment? So his assumption as an answer for this question was that only the ray that enters perpendicular to the eye affects the region, and the other rays are more refracted and therefore weakened. So most weakened signals or lights are not useful for the vision system or for the creation of sight, according to Al-Hazm, the Iranian scientist. But after a long period of time, after investing more on the vision system research, the fact is that the optical properties of the lens combine all of these rays into a single focused point under favorable conditions. Okay. The other important philosopher, researcher who lays the foundation for modern theories of optics and light is Jonas Kepler. He says light has the property of flowing or being emitted by its source towards a distant place. So this guy is intromissionist. That means he believes that light is originated and emitted towards the human eye from the external objects. And from any point, the flow of light takes place according to an infinite number of straight lines. He, came up with this idea called, or this fact called ray. That means each light travels on a straight line, and it can travel or it can advance to the infinite. So the lines of these emissions are straight and are called rays. So Jonas Kepler, he came up with the theories about optics. Optics is the study of light and also light. And he also introduced us the concept of rays as a light traveling in a straight line and with the capability to advance to the infinite. So the Kepler's retinal theory says even though light rays from many surface points hit the same point on the lens, then they approach the lens from different directions, and therefore they are refracted in different directions, separated by the lens. So according to Kepler's retinal theory, he says light is emitted from the external objects, as you can see here, from different external objects. It hits a medium called lens, and the lens is responsible to refract and collect or route these refracted lights to different coordinates in your vision system, so that we can create a sight. This is Kepler's theory of retina. When it comes to modern theories of vision, so far we have been talking about, the biological vision, of course, but the different views of vision by different philosophers or scientists. But when we come to the modern theories of vision, three main streams contribute to our understanding of vision. The first one is psychology of perception, more to the behavioral characteristics-wise definition of vision from the functionalities perspective. The second one is neurophysiology. That means a discipline that intends to explain how we see, how we create sight, or how sight is created. And the third one is computational vision. This one is more to representation of the vision system in general. So when we see the biological vision from two perspectives, the first one would be early vision. When we say early vision, it's the time, the early time when the human vision or an animal vision constructs something uh, useful for our brain system. So, for instance, under the early vision, we have parallelism, multiplexing, and partitioning techniques that are always implemented by our vision system in the early stages of vision. When we see parallelism, this is just to express that the human vision system, or in general, the vision system, is capable of processing multiple visual features at the same time, parallelly, simultaneously. The second one is multiplexing. Multiplexing is again another feature of our vision system at the early stage of vision that is used to explain or describe the fact that all those parallelly processed visual features are fused in order to bring something semantically feasible for our brain system. And the third one is partitioning. When we say partitioning, it's all about segmentation. It's all about identifying the distinct features of multiplexed features from the visual features from parallelly processed visual features. Okay. When we come to the high level vision, this is mainly about modularity. And when we say modularity, it's to emphasize on the modular structure of our brain system dedicated for specialized functionalities. For instance, face or facial perception has its own component or module in our brain system. Object recognition has also its own module or component in our brain system. So the biological vision system can be, classified as early vision phase and high level vision phase, as we already discussed. The early vision phase is more about parallelism, multiplexing, partitioning, and the high level vision mainly emphasizes on the modular structure of our brain for specialized processing of different percepts from the environment. This is the general structure or taxonomy of our eye. And here we do have a very important component in our eye system called retina. Retina is a place where photoreceptors are located, and different neurons like ganglion cells are also located. We do also have this component called fovea, where a high concentration of cones is stored. Around 50,000 cones are located here, but not no any rods. We'll talk about what photoreceptors are, what rods are, what cones are in a minute, but just to give you what are the components of our eye system based on this diagram. We do also have this thing called blind spot, where in our eye system where there is no um, rods and cones. So when visual features reach to this blind spot, then we will not be able to create some sight. So that's why we call it a blind spot. And there are different components here. For instance, when we extend the retina, we have rods, cones, and bipolar cells. So rods are mainly sensitive to light, so they are there for uh, mainly for perceiving in the dark or low level luminous intensity. Cones are there for spatial resolution. That means cones are more to precision and more to clarity or high resolution. So we are able to see during the daytime or the light because of the cones, and we are able to see um, during the night, even though it's with low resolution because of the rods. And bipolar cells are also called collector cells. These bipolar cells or collector cells are interconnected with the photoreceptor cells. Their main purpose is to filter visual features from rods and cones and transform them to the ganglion cells. Ganglion cells are again neurons in our eye system that is responsible to accept visual features from bipolar cells and transform them to our brain system. This diagram shows the details of how rods and cones are interconnected with collector cells or bipolar cells, and further how bipolar cells transforms visual features, filter visual features to ganglion cells. I will let you read this one in detail. This is more to the neuroscience and also biological aspects of vision system. But when we see retina, as you already said, we have three layers of cells. The first one is receptor cells, the second one is collector cells, and the other one is ganglion cells or retinal ganglion cells. So receptor cells are rods and cones. Collector cells are bipolar cells that are bipolar because they can accept from both rods and cones. And retinal ganglion cells are again neurons, which are directly related to our brain system. That means these components or these cells are responsible to transport visual features from our vision system to the brain. So when we see further the photoreceptor mosaics, the reason we say mosaics is that the two components in our photoreceptor component are rods and cones, which are naturally have different structure and purpose. Okay, so we usually say mosaics for some biological entities that is composed of two polar components. One could be negatively polarized component, the second one could be positively polarized component. Or you can also define the mosaics of something by the complete opposite functionalities of these components constituting that um, surface. For instance, in our case, the photoreceptor mosaics is defined by the combination of rods and cones. Rods are there for night vision, and cones are there for light vision. So for that purpose, we can say the photoreceptor mosaics to express the combination of rods and cones in our eye system, especially in the retinal subcomponent. So rods are approximately 100 million in our vision system, in our eye, and cones are approximately 5 million. As we already discussed, rods are sensitive to low levels of light, so we call these low levels of light scotopic light levels. And cones are sensitive to higher levels of light, and these higher levels of light are called photopic light levels. There are some light levels which are in the intersection point of scotopic and photopic light levels. We call them mesopic light levels. So at these mesopic light levels, both rods and cones are active. By the way, in the scotopic light levels, only rods are active, so cones cannot create sight at the scotopic light level. But when we come to photopic light level, cones are only active in this case. But in the mesopic light levels, at the intersection point or at the middle point between scotopic and photopic light level, both rods and cones are active. But there is the difference in conversion to receptor cells. You can see it here. So the other concept is pooling or conversion. So we said we have collector cells. These collector cells are responsible to accept visual features from both rods and cones, and we call this process pooling or conversion. It could be rod pooling or it could be cone pooling. Okay, so in case of rods, one important concept here is that several rods connects to each collector cell. That means it's more to many to one relationship. Okay, many rods connects to a single collector or bipolar cell. But when you come to cones and bipolar cells or collector cells have mainly one-to-one relationship, and it's limited connectivity. In case of rods, like all rods are connected to a single bipolar cell or collector cell, but when you come to cones, mainly cones have limited pooling or even connection, and so the pooling is also, the collection is also limited. Okay, cones doesn't give everything they have to the collector cells. This is due to, first, they have one-to-one relationship with the bipolar cell. So if you have one cone bipolar cell or one cone collector, then it's for fact connected to one cone. Okay, so they have one-to-one relationship. So the semantics behind this concept, the connection between rods and collector cells, cones and collector cells, is that rods are there to give us sight in low luminous environments. So they have to provide all they have to the collector cell so that the collector cell can filter and create some kind of sight. But when you come to cones, cones holds lights, so it's relatively easier to create sites with smaller lights than a large number of low luminous lights, right? So for that purpose, it's justifiable that cones have limited pooling, but rods have extensive pooling or extensive connection with the collector cells. So we can also refer to retina as a duplex. This is because of again the existence of rods and cones, the two modes of visual sensory. So we have this trade-off between sensitivity to light, that means being able to see in the low intensity, low luminous intensity environment, or spatial resolution, that means being able to see during the daylight or the light time when there is a light, and being able to have spatially high degree of resolution. So this is trade-off between rods and cones. For instance, when we come to rods usually trade um, um, sensitivity to light um, uh, for spatial resolution, and when we come to cones usually trade spatial resolution for sensitivity of light. Further on the duplex character of retina, we said rods have extensive convergence onto collector and ganglion cells, but we said cones have much limited convergence to these collector cells and ganglion cells. So because of that, cones have high resolution image of the world in good illumination, and rods have low resolution image of the world that persists even in low illumination condition. So when we further discuss or elaborate more on cones and color, there are three types of cones. We call them L, M, and S. L stands for long range wavelengths based cones. M stands for medium range wavelengths cones, and the second one is short, which is for short range wavelengths cones. Or we can also refer to them as yellow red cones, green cones, or blue violet or violet blue cones. So yellow red cones, it means these cones are able to see red type of colors or orange type of colors, and this is mainly referred to as long wavelengths colors. So basically these three different types of cones they differ in their sensitivity to different wavelengths of light, as we have already mentioned. So yellow red is more to long range wavelengths, green is mid range wavelengths, and violet blue is lengths, short range wavelengths. Okay, so these three components inside our cones are responsible to identify colors in three different wavelengths. For instance, if the object we are seeing is in the shorter wavelengths form, then we are able to see, the color in that short wavelengths, like violet blue. If the color we are seeing is, for instance, yellow red, then that yellow or red from the color perspective is defined to be in the longer wavelengths form. So the responsible cone for this would be L cone. Okay, the same is true for green or M cone, green cone or M cone. The other important concept talking about cones and color is that the concept of color blindness in low illumination. So we said cones are really good for spatial resolution, but their setback is that they are not able to create sight in low illumination. So cones are colorblind in low illumination. So if you don't have rods, for instance, then we are colorblind in low illumination, for instance, in nighttime or in a time where there is no light. Uh, Talking further on photoreceptor mosaics, there is also another component in our vision system called fovea, which is, as I already mentioned before, referred to as a place where there is a high concentration of photoreceptors. Um, but when we say photoreceptors, we are not talking about rods, but cones. So it approximately contains 50,000 cones in this area. So for this reason, the fovea cannot see dim light sources like stars when we look straight at them. The other topic that discusses the intersection between our cones, um, charge coupled device, which is device, an electronic device, and the space is this one. In this topic, the most important concepts are, for instance, it investigates the extent of the world the cone can see. Usually, the cone or the eye lens collects light over a total field of view of about 100 degree. Okay, and each cone collects light over a visual angle of about 1.47 by 10 raised to minus 4 degrees, which is about 30 seconds of visual angle. But when we come to CCDs, when we say CCDs, by the way, they could be they are charger coupled device, and they could be like digital cameras, HD cameras, or TV cameras. Different types of cameras or vision sensors are referred to as charge coupled devices. So when we come to TV camera, for instance, the TV camera photoreceptor mosaics, it's nearly square mosaic of approximately 800 by 640 elements, and this defines the complete field of TV camera photoreceptor mosaics view. So in terms of degree, for instance, the CCD, a single camera can see around 50-degree lens. Recall that the human eye can see about 100 degree. Okay, so this one gives 50 by 500, and this one means around 10 raised to minus 1 degrees per CCD angle of view. The other important concept on human vision is the blind spot. The blind spot is a place in your eye where there are no rods and cones. They are physical spots, by the way. So in order to know what blind spots are, I recommend you to use this diagram at the bottom. Try to close your left eye and focus on the white cross on the left side using your right eye. Then try to go towards and outwards the cross. And while doing this, try to notice the disappearance of the white circle to the right side. Okay, so here you see, for instance, if you see the red cross to the left side of the diagram using your right eye only, and if you go towards the cross just slowly, gradually, and outward from the cross, at some point in time you will lose sight of the white circle, which is at the right side of the diagram. So this shows the point or the time where the vision of this white circle hits the blind spot in your vision system or eye in general. So we call this spot a blind spot, and that means a human is not able to see anything when it intersects with its blind spot. So this is all for the biological vision. I mean, we can say a lot within the next 4 months about biological vision, but that's not our main scope. We used it as a foundation for the next lectures. But I recommend you to read more on human vision, biological vision, their intersection with computer vision. How does computer vision inherits some concepts or ideas or techniques from the human vision or even from animal vision? For instance, if you see infrared vision, it's inherited from other animals. If you see echolocation techniques of vision, it's inherited from another types of animals. Okay, so I recommend you to read about those things in detail, and for you to be encouraged for that, I propose a literature review, a simple literature review, but to the maximum of 3 pages literature review for you to undertake on topic related to like from human vision to computer vision, exploring the intersection of biological perception and artificial intelligence. So you can give it whatever name you want, but the domain should be in alliance with this domain. So with this literature review, we will dig more about human vision and how we advance to computer vision, and what uh, in what state of computer vision is right now, and what features or what techniques of natural vision are adopted for computer vision. I want you to dig more on that, study different literatures, and come up with a summary of concepts that reflects the path from human vision to computer vision, or generally the intersection of biological perception and artificial perception. Okay, that's all. The next topic will be about the camera system. I will also try to pre-record it and send it to you before our next class. Thank you.